on behalf of the Minas project team. So Miriam is a senior researcher at the University of Vienna, specializing in phylogenomics of human microbial pathogens using ancient DNA. And James is a postdoc that is shared between MPI-EVA and Leibniz HKI, both in Germany. He currently works and focuses on bioinformatics development for metagenomics with a bit of ancient DNA sprinkled in. He's very passionate about open source software, reproducible science, and increasing the quality of ancient metagenomics work with a collaborative and community-based approach. And yeah, today they're gonna guide us through this workshop on behalf of the Minas project team. So I leave you the floor.
Thank you very much, Bianca. So like I think both Bianca Maria and Maria have said previously, this is going to be an interactive workshop. This is not just you guys sitting listening. And to start off with that, I want to do some polling, which will happen if I can change my slides. So please raise your, I guess, virtual hand on Zoom, because otherwise I can't see anybody. Who has, for any of your projects, a master metadata table that has all of the samples and the libraries you're including in your project with associated metadata? Okay, about 2/3, not too bad. Who has had to, so next question, so you can put down your hands. Who has had to find metadata about sequences and from published articles or public data? So comparative data for your studies. A bit more. Nice, about half. Who has had to upload their own data to the ENA or SRA? Okay, much fewer. I feel sorry for the people in the future who are going to have to do that. And who has found any of the above, whether you've, regardless of which ones you have voted in, found that a nice, pleasant experience? Good. Okay, that has been the reaction to every single time I've done this sort of a question so far. So last question then is, who would like to have a standardized metadata table? You don't have to think about what goes into or anything like that. You just fill it in, and then it's the same thing that you can get from other places, from articles. So you go to a publication and you download the table, which has everything in the same format and so on. So most people agree with that, not everybody, which is strange, but okay, I'm going to go with that. So technically, actually, we are sort of halfway there. You can put your hands down now. So when it comes to publishing your data, genetics, I'm sure you're very familiar with, is very good actually at sharing data. Most people upload to the place like the ENA or NCBI SRA. And when you're uploading this data, you do actually have to fill in a certain set of metadata about the sequences you're uploading. And typically there's 3 different levels, which is the sample, experiment, and run. Which run basically corresponds to FASTQ files. And when you're doing this, when you're going through the submission process, you have to download a certain set of, you can pick which checklist you want to fill out, as you can see here on the right. So a lot of metadata is actually already available along with the sequencing data that you upload. And this includes stuff like mandatory things such as the sample ID, maybe the collection date and the sampling location, but also you can fill in with a lot of other things such as the isolation source. So what is the tissue type? What is the organism? What is the biological sex of that host organism? Even stuff like altitude or a level of solar irradiation. But I think that the last two may emphasize the point that you get some weird and wonderful things in these checklists. Some of them are so trying to pick which one can be difficult in the first place. And within there's a lot of extra stuff which is confusing or completely irrelevant to what you're working on, and you have to spend a lot of time trying to understand rather obscure or abstract definitions of these metadata tables, fields, and trying to work out what goes in there or not is really tricky. And leads this whole thing that you can spend a lot of time, and it's not the most enjoyable process, particularly when it comes to ancient DNA. Because, like I said, firstly, for ancient DNA, which one do you pick? If you open many of these, they're not really relevant to what you work on. I don't think I've ever seen ancient wastewater sludge. But also when you go into the ones that are more relevant, let's say the parasite checklist or the host-associated checklists, you will see many different fields which are completely irrelevant, and there's no way that we would ever know whether to fill in or not. For example, whether an individual had HIV or whether they had a lung or pulmonary disorder. Often in the archaeological sort of ancient DNA side, we're dealing with skeletons. You don't necessarily have that information because you don't have the soft tissue anymore. And so having to go through picking the checklist and trying to work out which other fields you want to fill in is very tricky. And so in an ideal world, what we'd rather like to have is a dedicated metadata checklist for ancient DNA sequences. So this means that you know that it will have relevant metadata for ancient DNA, things that are otherwise not covered currently. So sample age or damage treatment, things that are critical for our work, but often are just don't really apply to most other fields, so are not currently present. But also make sure that the requirements are applicable to ancient DNA. What I mean by that is not having, let's say, a mandatory term about something like the IHMC medication administration, whatever that says, code. Stuff that we'll never know, that just does not work, and that cannot be mandatory. We cannot apply to that data. So we need a different checklist which accounts for both stuff we are missing and also to account for stuff that we cannot infer from the past because of the nature of our data. And this is where the Minas project comes in. Minas stands for minimum information about any ancient sequence. And the ultimate goal of this is to be a standard and community agreed upon sample library sequencing metadata checklist for ancient DNA. And ultimately, more practically, what this means is basically a bunch of tables which are standardized that you can just fill in and then upload along with your publications, but also use that to fill in for data submission, a common sheet in many cases, which you can extend with your own stuff, but at least has stuff that is common across everybody. So it's very familiar to you and easy for you to pull in the information that you need and also upload the information you need. And they literally mean tables, just CSV tables, nothing even, no fancy sort of JSON schema or anything like that. And there are a range of benefits. So already we've seen from the polling at the beginning that often metadata reporting or data submission is quite of a difficult and boring process, which puts a lot of people off. So by standardizing, we're hoping this is going to make reporting easier for ancient DNA studies, because you just have a standard table you can upload to your publication. Because many of these checklists that I mentioned before already exist on the ENA or SRA, we hope by having an ancient DNA, specific one, will make it easier for you to submit data, because you can basically reuse the same table from your metadata upload and your ENA submission sheet. And also then, by extension, because all this metadata is in a standardized format, it's going to make it much more easy to find publicly available ancient samples and data on ENA, SRA, and so on. So let's say you want to pull more data for meta-analyses or to have comparative data for your study, let's say trying to get all the Yersinia pestis data, it will be much easier to find because you know there'll be certain keywords that will make it easier to retrieve this information. The long-term goal of the project is not just basically checklists, but actually trying to build some infrastructure around this, improving documentation, maybe build some metadata instruction tools, maybe things like validation tools or metadata APIs and things like this. And also at the same time, parallel, want to provide training. So particularly when you get to data upload to these repositories like the ENA or SRA, the documentation from these archives are often quite poor. And it's something that no one really considers to the last minute of the project, and poor students have to just work it out by themselves, which is not really fair, to be honest, when it's such a actually critical part of particularly the open science movement nowadays. And so we want to provide maybe training and show different ways of uploading and so on. And the way we want to do this is, like I said, is these checklists, these CSV sheets, which actually are coming from this existing consortium called the Genomic Standards Consortium. They have actually written many of the checklists you already get on the ENA SRA and have been adopted in many different fields. You can see actually they've been very successful with this. If you're into impact factors, if that is important to you, you can see that most of the main ones have been published in Nature level journals and quite high level tier ones, which shows at least that the editors of these journals think this is a sexy topic and an important topic. when normally they're just chasing headlines. And so the Genomic Standards Consortium has a lot of infrastructure, which we're basically building upon, and they're the people who sort of facilitate getting all of our metadata and then standardizing into a way that we can get it onto our different repositories. But like I said, this is not fixed. It's not just for uploading metadata, these sample sheets. They are also usable in many different contexts. So even if you yourself don't expect to upload your own data, you can still use the same checklist in other contexts as well. So like I said before, the first goal of Minas is to basically extend the existing metadata checklists by adding something what we call a MixS extension. So this is sort of so many of the checklists from the GSC, the Genomic Standards Consortium, are modular. So for example, you'd have something like a metagenome plus host associated, which I mean is something from a body. You can also have human associated, or you could have a metagenome plus sediment DNA checklist. So we want to make one of these modules, which is just for ancient sequences, which means that is the metadata that only us in ancient DNA would ever care about, let's say damage treatment. And then the second goal of Minas is to develop submission packages or combinations. And this means is actually building this combination of modules. So let's say the metagenome, host associated, and then plus this ancient package. So we have this as one single checklist, which you can then adopt for, let's say, microbiome research or pathogen research and so on. So these are the two main goals of this current phase of the project. And so what we have asked you to come here today is to actually contribute to the development of these checklists. We want to discuss the ancient DNA specific metadata columns and fields. So we want to actually ask, what would you like to be included in such metadata things? Let's say when you're trying to search for data online, or whether you're trying to fill in your own information for your own checklists. We have our own proposals, and what we're going to do in the interactive session of this is get all of you guys to go through and simulate trying to fill in such a checklist, and then you give us the feedback of how that was, or what our drafts already give you, but also have in mind your own ideas of what you think is missing that we could incorporate, and you can give these ideas back to us. And so the ultimate sort of, to summarize, the reasons why we feel we want the community to get involved and why it's important that you do give your feedback is for a variety of reasons. Firstly is that for it to succeed, it requires community consensus and buy-in. So if we unilaterally decided this is what the information that's going to go on these checklists, most people say, no, we don't agree with it for XYZ reason, we're not going to use it anymore. So if everyone agrees, roughly, so there's a consensus, it means that people are more likely to buy in to the project to make sure it works. We also need a diversity of feedback, whether that's students from masters or bachelors all the way up to sort of PIs, but also people like archaeologists and museum curators as well, to get their feedback here, again, leading back into consensus stuff. And to sort of justify this and your time, we have a carrot and a stick approach here. So the carrot is that if you do provide feedback here, so contribute to the project, and pledge to use the eventual sort of outcome of the project, we'll offer basically co-authorship in the Minas publication. This sort of hopefully serves two purposes. One, authorship is the currency of academia at the moment, but also by having your name on the publication, it would sort of also put a stamp of show that you want to sort of commit to using it. But also the stick, or baguette in this case, because I couldn't find a stick emoji, weirdly enough, is that if you don't give your feedback, that's fine. You don't get involved, that's also fine, but you will have to use the checklists anyway. So if you want to have your say, you better use it now, because once it's on the ENA, it's going to be much more difficult to change this and so on. This is actually a multi-year project. It doesn't end at the end, but once we have the main draft, that is pretty fixed. It's going to be more complicated to sort of review and update over time. So it's better to have your say now when we're in the sort of fun creation phase. So the task, which we're going to do now for the interactive session of the SPAAMtisch, if you're willing to accept it, which I hope you are, is ideally in groups of 2 to 3 people. We would like you to take the draft checklist, which I'll show you how to get to in a second, of your choice, so most relevant to your work. Take 2 to 5 of your current projects or last publication, if you've already published something, samples, and just try filling in the checklist. And when trying to fill in this checklist, we want you to be noting down things such as, what is your general experience? Do the definitions of the metadata terms work or not? is there any metadata you think is missing? So specifically for, let's say, ancient DNA, is there a critical bit of information that does not exist that you shouldn't think should be reported by anybody, any other researcher? And also consider, as I'll show you in a minute, whether the field should be mandatory or optional. And sort of, we'll give you about 60 minutes, I think, to try and do this. And then we'll have basically a feedback round after, where we just ask for your opinions. In like a round table sort of system. So we'll ask you to say what are the major issues and see if other people agree or not. So the first thing is then actually please download the relevant draft checklist for your research spreadsheet. So maybe Miriam, you could, if you're there, or Bianca Maria or Maria, copy and paste this link if you can get to it into the chat. And what you'll see is a table, as you can see in this screenshot here. Try and pick the one that's most relevant and download the Excel sheet that will be listed next to it. So I'll give people, let's say, 30 seconds to do that. Once the link is in the chat, hopefully.
It's in the chat.
It's in the chat, thank you. I should also, we're focusing on just sequencing reads at this point. So theoretically, the sheet can be extended to things like submitting metagenomic assemblies or actual consensus sequences, but we're not going to cover that today because it's a sort of takes a different approach to get around that. So just focus on maybe the top 10-ish sheets.
Once you've chosen them, you can actually leave a comment on the link to tell me which one you chose, so we have an idea of which one you're working with. That'd be great.
That would be, yes, please. Okay, I hope everyone's downloaded it. If not, the link is in the chat. Once you've downloaded your particular checklist, and please only pick one, don't try to do multiple. What you will find inside is 4 tabs. One is called Legend, which is basically your key of what each metadata field corresponds to, so the description of each entry. The requirement levels, so the GSC has this level of mandatory, optional, not necessary, and then environmental dependent levels of how important it is and whether it's mandatory or not. There's a description of that key in that tab. There's a version tab. That doesn't really matter. It's the version of the checklist you're looking at. And finally, the data tab. Now, this is the important one, and this is the one we want you to basically try filling in. And what we basically want you to do for the next, about 60 minutes, ideally, is for each row of the data tab is to read what the metadata entry means, what you're meant to fill into it. So basically check the legend tab. Fill in one of the columns that's prefixed with sample in that column of the data tab. And then as you're going through it, if you're having problems or suggestions or idea, please leave that in the notes section. This can also just be like, I don't understand this whatsoever. Like, that's also fine. And then if you can get through that in an hour for one sample, please try this again for a second sample, just to make sure that you can get an idea of an average of the experience of this. When you're doing this, we're going to actually split you into groups of 2 to 3 people. So again, we want to get your experiences as in a simulation of you pretending to be someone who's uploading data, for example, at the end of a publication. So we'll split you into 2 to 3 groups, into Zoom rooms, groups of 2 to 3 people. If you really don't want to be in a group, that's fine. Please just tell Miriam. Then please discuss. If you get stuck, please ask within your groups. If none of you can work out what it may mean, then just skip that entry, leave that comment in your notes, and then just carry on to the next entry. And finally, keep in mind, while you're going through this, always think, what is the metadata you think is critical for your work that any ancient DNA person with either in your subfield or generally in ancient DNA should be filling in, and check if that's there or not, because it could be that it's missing, and if so, we should add it to our draft checklist. So that is your task. This is sort of a very, very basic example of what maybe it would look like at the end. So each row is a different metadata entry. As you see, project name, sample taxon ID, ethical authority. You'd be checking these against the legend tab if you don't know what it means. the requirement levels, whether it's mandatory or optional, etc. You then start filling it in as much as you can. And then in the notes, if you have a question or you don't understand something, you can leave it here in this bit there. So hopefully that sort of roughly shows you what it means. Then, like I said, after the task is complete, we'll basically have a roundtable Q&A. You can basically bring up what the issues you had. You can think about like what are the 5 major terms that you found difficult, didn't understand. Also think what are your ideas, what you think is missing, and so on. And then at the end, if possible, it would be nice if you could share with you your filled-in sheet with your notes. If it's from unpublished data and you can't share it, that's also fine. Don't worry, but just if you can, that'd be great. And also, please let us know if you would like your name to be included in future publications if this goes forward. Generally, we'll be noting who gives feedback, and obviously, just by attending, you're not going to get co-authorship. But if you give useful feedback, then you can be included there.
If you send us your sheets and you don't want to share your data, you can also remove the data before sending it with just the feedback, but just make sure the feedback is understandable without us having to see what you filled in, if possible.
Yes, exactly. That's another way around it, indeed. And so just before we start, then just say, if you're interested in the project as a whole, we have a mailing list. Oh, that link is probably dead now, but it should be under the, I can't remember what it is now. 1 second. I should update that. Get involved. If you go to get involved, we have a new mailing list, what I said last week. You can join here. Then we have a chat. We are on Slack as well, on the GSC chat. So press the right button. And also a lot of the discussions of the terms will be happening on GitHub, so you can trace that over time. And with that, we'd like to thank you to everyone who's contributed so far. And I hope to see your names on this in the future. And then thanks to GSC and SPAAM where the idea came out from. And then my half, there's MPI-EVA HKI, my employers, and the Vienna Stiftung. And for Miriam, the University of Vienna and the Austrian Science Fund. So thanks to them. But otherwise, before we start, do you have any questions? Does everyone have a sample sheet downloaded? Oh, sorry, does anyone not have a sample sheet downloaded? That's a better way of asking. I'll take that as a no. Then I would maybe ask the Zoom rooms to be set up.
Yeah. We have 4 breakout sessions right now. Since I think a bunch of us are organized as well, but I think we can fill like 3 of them, I guess, with like 2 to 3 people per. So if you could all just join one for a discussion, we'll be popping in every like 15 to 20 minutes to see if everything's all right. Like James mentioned, this isn't really a workshop in the sense that we're teaching you how to do it. This is you teaching how we're doing it wrong to some degree. We want to know what doesn't work for you, what confuses you. So actually you telling us, okay, this doesn't make any sense to me, is actually way more helpful for us. If you could also, include like in my field, what that field is, and maybe put a specific example of how this could be complicated in the comments as well for us. Make it so we can have an understanding of the issue as much as possible from the actual sheet once you share it to us. But I will be also going through a discussion at the end after the hour, and we can discuss it as a group, for any bigger issues that came up or any term you want to add that's not present, like you are doing something really niche in your research and there is a term that you always need and that might be relevant to more people than you, and that's not currently in the list. That would be something we also would want to know.
Okay.
I've also shared the slides in the chat. So in case you have any questions, you can check that at any point. Otherwise, I'll hand it over to Bianca and...
Yes, no, I just wanted to say that I think so the rooms should be open so you can join them now. Of course, if you see that there is already 3 people in there, like you can come out and change another, they can change and go to another room. So yes, you can go to the rooms. Now, I think if you see them, if you have problems in seeing them, let me know.
Otherwise, thank you and see you in about an hour or 15 minutes.
I'll stop the recording.